Bismillahirrahmanirrahim. My name is Dr. Imran Waheed. Today I will be discussing the physiology of urine formation. Those of you who have not listened to my previous lectures, for them I will tell you that the urine formation is a process of three processes mainly. Urine is formed by filtration, reabsorption and secretion. These are three processes which are involved in urine formation. Filtration occurs at the level of glomerulus, reabsorption occurs at the level of tubules, proximal, distal, convoluted tubules, and secretion occurs at the level of tubules. Actually, what happened is that so far as filtration is concerned, all of us have learned in the previous lectures that the blood circulates in the glomerulus and the resulting fluid which is present in the urinary space of Bowman's capsule is known as glomerular filtrate or plasma filtrate. Many important ions, large molecules, blood cells or amino acids and proteins which do not cross the glomerular basement membrane or filtration barrier, they are not filtered and they are retained in the blood. This glomerular filtrate is actually blood plasma without blood cells, proteins, RBCs, WBCs because of their large size, because they cannot cross the filtration barrier which is formed by the endothelial cells of the glomerular capillaries to epithelium of the visceral layer of the Bowman's capsule which has octopus like foot processes and they are known as podocytes. So, there are three components of the filtration barrier or glomerular filtration barrier. Number one is the endothelium of the glomerular capillaries, number two a basement membrane, and number three the epithelial cells of the visceral layer of the Bowman's capsule which are known as podocytes, and they have octopus like foot processes embedded in the basement membrane. So, this is the brief introduction about the glomerulus. Before I delve into the details of the urine formation, let me tell you another important thing. We will see in this brief diagram. This is a line diagram very nicely, beautifully explaining various processes of urine formation. So, this is a line diagram of Bowman's glomerulus. This is the artery, and this is the tuft of capillaries or glomerulus. This is the Bowman's space. This is the proximal tubule, and this is the distal tubule, and this is the excretion part of the urine. Here we can see that the glomerulus, this is the blood end, arterial end, the peritubular blood. So, on the one side there is tubule and on the other side is peritubular blood. So, this is important to understand that blood circulates in the glomerulus and the filtrate which is formed is known as glomerular filtrate, the composition of which I have told you. So, the composition of the blood filtered in the glomerulus or glomerular filtrate is basically containing water, electrolytes like sodium, potassium, etc., small molecules such as glucose, amino acid and waste products like urea. These are the components. There are certain sequence of events as the fluid is filtered and it moves out of the glomerulus into the urinary space of the Bowman's capsule and enter the proximal tubule and travels down its distance from the distal convoluted tubule, collected it and finally excrete it. Approximately 99% of the glomerular filtrate is water. The concentration of ions like sodium, potassium chloride and others are present in similar amounts as in plasma. It is normally present in the filtrate. So far as glucose is concerned, glucose is normally present in the glomerular filtrate. Similarly, amino acids are also present in the glomerular filtrate. A nitrogenous waste product like urea is also present in the filtrate. Uric acid, which is the waste product of protein breakdown, is also present in the filtrate. In other words, we can say that products such as serum creatinine, including some other small molecules, are also present in the glomerular filtrate. So, basically it contains water, electrolytes like sodium, potassium chloride, bicarbonate, glucose, amino acids, urea, creatinine, uric acid and other small molecules. Now, the question arises, what are the changes which take place in the tubular lumen, in the tubules of the kidney, proximal, distal, convoluted tubule, loop of Henle, which convert this glomerular filtrate or plasma, filtered plasma, into a urine. The pH of the blood is alkaline, that of the urine is acidic. The urine is sometimes concentrated, sometimes dilute. What are the changes which take place in the glomerular filtrate filtered by the glomerulus, which travel down its journey from the Bowman's urinary space to the excretion in the collecting ducts? What are the changes which take place and is converted into urine? So this is a line diagram which shows Bowman's space, proximal distal convoluted tubule, collecting ducts and excretion, and on the other end there is glomerulus and the tubular peritubular capillary end. The excretion of the substances as they pass in the urine. So this is very important to know that this was filtration actually. 180 liters per day of the filtrate is formed, 125 ml per minute. Total plasma volume is about 3 liters. So this gives us some idea of how often plasma has to be filtered and reabsorbed. In proximal convoluted tubules, the process of reabsorption takes place. Reabsorption is the process by which substances from tubular fluid pass to the surrounding peritubular fluid and then peritubular blood. This is the peritubular blood. The third phenomena is secretion. This is the process by which substances from peritubular blood pass into the tubular fluid. So the filtrate which is filtered from the glomerulus in the urinary space of Bowman's capsule, traveling through the proximal tubules, loop of Henle, distal convoluted tubule and collecting ducts, there are some elements are reabsorbed, some molecules are secreted, water is reabsorbed, and it results in the formation of urine, which can be concentrated or dilute depending upon the needs of the body. So this is very important to know that the reabsorption or the second process, this takes place in the this urine formation process. Second process of reabsorption takes place in the renal tubules. The function of renal tubules is basically to accomplish this second step in urine formation. As the glomerular filter passes through the proximal and distal convoluted tubule, loop of Henle, distal tubules, and many essential molecules and ions are reabsorbed in the peritubular blood, while some of them are secreted in the tubular fluid, as I mentioned, from the blood. So as we have discussed previously, that glomerular filtrate produced as a result of glomerular filtration, it undergoes further changes as it passes down its journey from proximal tubule, loop of Henle and distal tubules. So following are the reabsorption and secretion processes which take place in the tubules. That is, the fluid which was filtered by glomerulus is now known as the tubular fluid. So we must know that which mechanisms are basically involved in movement of ions from the peritubular fluid into peritubular blood into the lumen of the tubule, and from the tubular fluid into the blood capillaries, blood of the capillaries. So there are four main mechanisms. Number one, some ions are moved by active transport. The second one is passive transport or simple diffusion. Third one is the exchange pumping or counter transport. And fourth one, last but not least, is the movement or transport across a potential gradient. So it is very important to understand that these processes they keep on going in a smooth fashion, so that those elements which are needed by the body, they are retained, and those which are not needed by the body and waste products are removed from the body. So now we will see that how this process continues. So this is another diagram I will show you. This clearly indicates and describes the process of tubular reabsorption and secretion very beautifully, and we can understand how the whole process of reabsorption takes place. But before this, once again I would like to tell you that this is very important to understand the basic mechanisms which are involved in filtration. So due to the filtration barrier in the glomerulus, large molecules like RBCs, WBCs and plasma proteins are excluded from the glomerular filtrate because they are unable to cross the glomerular filtration barrier due to the large size. So what are the changes which actually take place? following changes take place in the glomerular filtrate as it passes down the proximal and distal convoluted tubules and collecting ducts and loop of Henle obviously. We can see here that in proximal tubule you can see 60% of the water, 60% of the water is reabsorbed, which was filtered by the glomerulus. 65% of the water which was filtered and sodium is reabsorbed. Due to this, the fluid having the fluid leaving the proximal tubule to enter into the loop of Henle is virtually same as that of plasma. That is 65% of sodium and water is reabsorbed in the proximal convoluted tubule. Sodium is absorbed by active transport and water by simple diffusion. Similarly, we can see that potassium is reabsorbed, hydrogen is secreted, glucose is reabsorbed, amino acids are reabsorbed, uric acid, vitamin C, organic acid and lactic acid. This is very beautiful description in proximal convoluted tubules. Chloride moves by simple diffusion, water by simple diffusion, carbon dioxide, urea and protein by simple diffusion in proximal convoluted tubules. So, the fluid now which is leaving the proximal convoluted tubule to enter the loop of Henle is basically as same as that of plasma. In first half of the proximal convoluted tubule, sodium is reabsorbed. Sodium is reabsorbed by co-transport with bicarbonate. Co-transport with bicarbonate, while glucose, amino acid, phosphate and lactate are also reabsorbed. Glucose, amino acid and lactic acid is also reabsorbed. In the second part of the proximal convoluted tubule, there is a high concentration of chloride. It diffuses passively through the tight junctions. Chloride diffuses passively, this is simple diffusion through the tight junctions into the intercellular spaces, making the basal side of the cells negatively charged with respect to the tubular fluid side, due to which sodium follows the chloride ions across the tight junctions into intercellular spaces along this electrical gradient. So, movement of reabsorption of sodium, potassium, hydrogen, glucose, amino acid, uric acid, vitamin C, organic acid and bases and lactate in the proximal tubule by sodium, potassium by active transport, hydrogen is reabsorbed and organic acids are also reabsorbed, while chloride, water, 65%, carbon dioxide, urea and protein are reabsorbed by the process of simple diffusion. So, this is very clear and important that we can understand the reabsorption very easily by this diagram. Now, we enter the descending limb of the loop of Henle and we see that in loop of Henle, the remaining 25% of the sodium is reabsorbed by the loop of Henle. The thick ascending limb is very important regarding the sodium transport. In thick ascending limb, the mechanism involved is co-transport with 2 chloride and 1 potassium ion, that is symport. It is also counter-transported with hydrogen. Counter-transported with hydrogen. You can see here. So, to summarize, 99% of the sodium filtered from the glomerular capillary is reabsorbed as the tubular fluid passes down the nephron. The reabsorption depends upon the active transport of sodium potassium ATPase mechanism, which pumps sodium from the basolateral membrane of the tubular cells into the peritubular fluid. Similarly, the intercellular concentration of sodium is therefore kept low. So, this is very important that the tubular fluid, the concentration of sodium is high, it moves across the apical membrane into the cell down the electrochemical gradient. So, if we compare the composition of capsular filtrate, which is filtered in the Bowman space, the filtrate fluid which is formed from the glomerulus, the glomerular filtrate, if we compare the composition of this filtrate with fully formed urine, there are some differences between the glomerular filtrate and fully formed urine. In 24 hours, the total volume of urine is much less than amount of capsular fluid. 170 liters of filtrate are formed by the glomerula in 24 hours. Only 1.5 liters of urine is excreted daily. There are some substances that are present in the filtrate, but are practically absent in the urine, for example, glucose and bicarbonates. The solid components are much more much more concentrated. The solid components, they are much more concentrated, and in the urine as compared to plasma or filtrate. All these, the solids, they do not undergo the same degree of concentration. Each of them have a different concentration in urine. The total quantity of some solid substances in the urine is much more than that in the capsular filtrate. For example, the pH of blood, plasma and capsule is alkaline, same as plasma, but the urine is acidic in reaction. Some substances are present in the urine which are not present in the capsular filtrate or renal artery. Now we will see how these changes take place in the capsule filtrate, converting it into urine. It is obvious that these changes must have taken place in the capsular filtrate when it passes down the renal tubules. Actually, the function of these renal tubules actually is to bring about these changes actively in the capsular fluid and thus convert the capsular filtrate into fully formed urine. These changes which convert the capsular filtrate into fully formed urine during its course through the renal tubule is accomplished by selective reabsorption and tubular secretion, and formation of some new substances is also involved. So we can see here that amino acids and proteins, normally a small amount of protein is filtered into glomerulus and is reabsorbed through the brush border of the peritubular epithelium by pinocytosis. Similarly, the glucose under normal conditions, glucose is filtered. It is also completely absorbed in the first half of proximal tubule by the active transport. Water, as I have mentioned, out of 170 liters of water filtered by the glomerulus of the kidney, 168.5, that is 99%, is reabsorbed in the renal tubules, and only 1.5 liter is excreted in urine. Water is reabsorbed in proximal convoluted tubule, the distal convoluted tubule and collecting ducts. Normal osmolarity of plasma is 280 milliosmoles per liter, which is maintained by the kidney ability to excrete water and sodium. Proximal convoluted tubule has high permeability to water. About 2/3 of the or 7/8 of the water of the glomerular filter is reabsorbed in the proximal convoluted tubule. Active reabsorption of sodium along with passive reabsorption of water takes place in the proximal tubule. Here water and electrolytes are reabsorbed as an isoosmotic solution, but do not play any role in concentration of the urine or dilution of the urine. This is called obligatory water reabsorption. That means the water is being absorbed by obligatory water reabsorption method. Sodium and potassium are reabsorbed in proximal tubule by active transport, but chloride is reabsorbed by passive diffusion. Water is also reabsorbed by passive transfer or diffusion. Due to these changes, the tubular fluid which is leaving the proximal tubule is isotonic. Now, what happens with this isotonic fluid? That in loop of Henle, we can see in loop of Henle that the ascending limb of the loop of Henle is impermeable to water. The ascending limb of loop of Henle is impermeable to water. Of the loop of Henle maintains the same osmotic pressure as interstitial fluid, and water and sodium pass by diffusion into and from interstitium respectively. The descending limb of loop of Henle is permeable to water. Descending limb of loop of Henle is permeable to water, whereas the ascending limb of loop of Henle is impermeable to water, and it is here that sodium is pumped out actively into the interstitium, so raising the osmolarity and providing a hyperosmolar medium which allows the loop of Henle act as a counter current multiplier system, which we will discuss later on. This is the active reabsorption of sodium by active transport, due to which the tubular fluid becomes dilute and hypotonic. The fluid which left the proximal tubule was isotonic, but now the fluid which is leaving the loop of Henle becomes dilute and hypotonic. So the tubular fluid leaving the loop of Henle is hypotonic. Now we come to the distal convoluted tubule. In distal convoluted tubule, we can see that the tubular fluid undergoes changes, that is epithelial cells of the distal convoluted tubule can establish a relatively high ion concentration gradient between the blood and tubular fluid, which is actively altered with the change of blood content of ions, which is sodium and potassium, and it is also controlled by mineralocorticoids like aldosterone of the adrenal cortex. Distal convoluted tubule performs an other function by establishing relatively high osmolar concentration gradients between blood plasma and tubular fluid, which undergoes changes by percentage of body fluid or water content. The water permeability through the epithelium of the distal convoluted tubule is affected by the circulating levels of antidiuretic hormone, which is secreted by the supraoptic nucleus of the hypothalamus. This hormone causes significant reabsorption of water by the distal convoluted tubule. There is significant reabsorption of water by the distal convoluted tubule. We can see in the previous diagram that there is significant reabsorption of water by the distal convoluted tubule. Here we can see this is the distal convoluted tubule. There is significant reabsorption of water. You can see here significant reabsorption of water under the effect of antidiuretic hormone. Here is the antidiuretic hormone and it causes significant reabsorption of water. So this is very important to understand. So the secretion of ADH hormone is controlled by water content of the body. To summarize, we can say that the reabsorption of water in the proximal tubule and in the loop of Henle and distal tubules is a passive process, that is diffusion, which is determined by the osmotic forces and gradients created by the reabsorption of various ions. Now we come to the collecting ducts. In collecting ducts, the tubular fluid which is leaving the distal tubule now enters the collecting ducts, which is permeable to water. This passes through the walls of the collecting ducts with the osmotic gradient into the medullary interstitium. It is at this stage that ADH exerts its effect and enhances the movement of water, that is reabsorption, producing a hyperosmolar fluid or urine. So here the osmolarity 400, 600, 800 osmolar. So the fluid which left the proximal tubule was isotonic, which left the loop of Henle was hypotonic, and in collecting ducts we can see that it is hyperosmolar urine or fluid. ADH is therefore acts at the distal convoluted tubule than collecting ducts to cause significant reabsorption of water. So this is very important, that reabsorption of water is responsible for concentration of dilution of the urine by itself as well, thereby increasing the osmolarity of the tubular fluid. Reabsorption of water increasing the osmolarity of tubular fluid, resulting in hypertonic fluid or urine, which is hyperosmolar to plasma. More salts and ions as compared to water are absorbed in the loop of Henle as hypertonic fluid is entering into the distal convoluted tubule from loop of Henle. Under conditions of hydropenia or water deficiency, water is reabsorbed quickly across the distal tubule's epithelium, which becomes permeable to water. Urine is not concentrated in distal tubules but in the collecting ducts. In distal convoluted tubule, important thing to understand is that there is reabsorption of water which is regulated by ADH, as we have discussed. This type of absorption affected by ADH is known as facultative water absorption. This is active reabsorption of sodium and chloride as well in the distal convoluted tubule, due to which the tubular fluid becomes isotonic again. In collecting tubule, this isotonic fluid becomes concentrated, as I have discussed earlier, due to further reabsorption of water. So the reabsorption of water occurs as follows. In proximal convoluted tubule, 2/3 is reabsorbed, which is known as obligatory water reabsorption. In distal tubules, water is reabsorbed under ADH, and this is known as facultative reabsorption of water. In collecting ducts or tubules, the reabsorption of hyperosmotic fluid occurs. It is very important to understand that the reabsorption has to work against the gradually rising osmotic gradient or pressure. It is obvious that a point will come where these two opposing forces will equalize further and reabsorption will stop. It means that there is the maximum limit beyond which the urine cannot be concentrated. The solid constituents of urine therefore need a certain minimum amount of water for excretion. This phenomena is known as minimum or obligatory volume. Now, before we leave the topic, we should see what is counter current multiplier mechanism, which is responsible for concentration of urine. It is quite evident from our discussion that pH of the blood is 7.4, which is alkaline, but the urine is average 6, which is acidic. The glomerular filtrate is fluid which is isotonic to plasma, which means that it has similar solute concentration. It moves down the nephron, the proximal tubule, loop of Henle, collecting tubules. Its tonicity changes due to reabsorption of water and solutes. The osmolarity, which is measure of solute concentration of initial glomerular filtrate is formed, is same as that of plasma, 275 to 295 milliosmol per kg. Osmolarity is actually measured of a solute concentration. It is a measure of solute concentration. The different phases through which the urine becomes hypertonic and concentrated as it moves down the nephron after leaving the glomerulus and entering into the tubules is important. It is a complex process which is closely related to the anatomical distribution of tubules, along with sodium concentration at different depths of the cortex toward the medulla of the kidney. The loop of Henle of juxtamedullary glomeruli forms loops. They form the loops which dip deep into the medullary cortex, and here we can see them. This diagram is very important. They dip deep into the medullary cortex. Loop of Henle dip deep into the medullary cortex, and this is the descending limb. This is the descending limb, this is the ascending limb. They dip deep into the medulla of the kidney, accompanied by the blood vessels. These blood vessels arising from the efferent arterioles of the juxtamedullary glomeruli, they also dip down into the kidney to form loops of straight vessels, and then they return to the cortex. The loops of Henle of juxtamedullary glomeruli, they dip deep into the corticomedullary, from corticomedullary junction to medulla, along with the parallel vessels which also form loops and are known as vasa recta or straight vessels. They are parallel to each other. This is descending limb, ascending limb, descending limb, ascending limb. Similarly, the blood vessels are parallel to them. It's very important to understand the mechanism of formation of concentration of urine. Very important to understand the concentration of urine. This is the fluid in the proximal tubule, cortex, medulla, and this is the distal convoluted tubule and collecting ducts. Here we can see that the concentration of fluid which was filtered takes place, and these are the vessels which I was talking about, the vasa recta. These are straight vessels. They also form loops parallel to the loop of Henle. Juxtamedullary glomeruli, efferent arteries giving rise to this loops of vasa recta. So, moving back to same, we can see that in the descending limb of loop of Henle, ascending limb, this is blood and the fluid in the lumen. They return toward the cortex, vasa recta, and form loops. They look like a hairpin or counter current management. Hairpin or counter current management is the word which should be remembered to understand these important changes. So, counter current mechanism is responsible for the concentration of the urine. This is the diagram which will make us understand what is the counter current mechanism. These are vasa recta and this is the loop of Henle. So, it acts like a hairpin counter current management system. Following are the changes which take place during the counter current multiplier changes resulting in urine concentration. The glomerular filtrate have a concentration same as that of blood plasma. About 80% of the filtrate is reabsorbed in the proximal tubule. The remaining 20% enters into the descending limb of loop of Henle as an isotonic fluid. When this as isotonic fluid enter the descending limb of the loop of Henle, there is passive diffusion of sodium into the tubule from the surrounding hypertonic tissue and fluid becomes hypertonic. The osmolarity starts increasing. This is clear here. Now, this hypertonic fluid enters into the ascending limb of loop of Henle and gradually becomes hypotonic due to active reabsorption of sodium from tubule to the surrounding tissue and also due to impermeability of ascending limb of the loop of Henle to water. This hypotonic fluid enters into the distal tubule, where antidiuretic hormone increases the permeability to water, resulting in again in isotonic state. Finally, this isotonic fluid passes through the collecting tubules and becomes hypertonic due to ion and water exchange between the tubular fluid and hypertonic medullary interstitium or tissue fluid, resulting in formation of hypertonic urine which passes out through the ureter. So, this is the important point to understand that these are loop of Henle and these are vasa recta. Changes in solutes take place which give rise to increasing osmolarity and blood plasma which is isotonic is converted to hypertonic urine. It is a counter current exchange is basically a passive process and depends upon the diffusion of solutes and water in both directions across the permeable walls of vasa recta. It takes place in both directions. You can see entering into the vessel and leaving the vessel. As the descending limb of vasa recta gradually enters deep into the medulla, water diffuses out from and osmotically active particles diffuse into the blood vessel. In the ascending limb of loop of Henle, along just like that, in ascending limb of vasa recta, this diffusion process is just in the opposite direction. As thus the isotonic blood is leaving the medulla and this counter current exchange reduces the rate of dissipation and hence reduces the rate at which the counter current multiplier must pump sodium to maintain any given gradient. Sodium is reabsorbed along with chloride. Out of 560 grams of sodium which is filtered in the glomeruli, 490 grams are reabsorbed in the proximal tubule and 65 grams are reabsorbed in the distal columnar tubule, while 5 grams are excreted in urine. Reabsorption of sodium is an active process in tubule due to active transport in the cells of ascending limb and distal columnar tubules. In descending limb of loop of Henle, the movement of sodium enters the tubular lumen by process of diffusion. That is in the opposite direction. Reabsorption of water follows passively due to active reabsorption of sodium in the tubule. This absorption is influenced by aldosterone which causes sodium retention and potassium excretion. So far as potassium is concerned, as I told you, 80 to 90% is reabsorbed in the proximal tubule and a specific active transport process is involved in this reabsorption. The secretion of potassium takes place in distal columnar tubule. This is the distal columnar tubule and here the secretion of potassium takes place. Here we can see the potassium is secreted in the distal convoluted tubules. Secretion of potassium in the distal convoluted tubule. Here it is shown in distal convoluted tubule, secretion of the potassium. Again coming to this diagram, we can see that bicarbonate is also completely reabsorbed in the renal tubules. Bicarbonate is also completely reabsorbed in the renal tubules. Here we can see bicarbonate is completely reabsorbed in the renal tubules. Phosphate is reabsorbed in the proximal tubule. Phosphates and reabsorption of phosphate is an active process which is affected by the parathyroid hormone. Uric acid, sulfates, ascorbic acid, beta hydroxybutyric acid, acetic acid, creatinine and some amino acid, they are also reabsorbed by the proximal columnar tubules. Third and final step in urine formation is tubular secretion. So, this diagram will also tell us about the tubular secretion. Tubular secretion is very important. Here we can see, although the selective reabsorption is the main function of the renal tubules, but they also secrete some substances. Tubular secretion is an active process carried out by the tubular cells. Potassium, creatinine and hydrogen ions are also secreted by the tubules. When sodium ion is reabsorbed in the tubules, sodium hydrogen ions are at the same time potassium and hydrogen ions are also absorbed secreted at the same time. It means that sodium is being reabsorbed in the tubule, while potassium and hydrogen ions are at the same time being secreted in the tubules. So, tubular secretion is another process. When the sodium reabsorption is increased by aldosterone, it is retained, potassium is secreted, which is this secretion is increased by the effect of aldosterone, sodium retention and potassium secretion, which is excreted in the urine, and when the secretion of hydrogen ions increases, that of potassium decreased. This was the brief introduction to the physiology of urine formation. If you concentrate on this diagram and listen to what has been said carefully, I think you will understand the physiology of urine formation. It is a difficult topic. If there are any queries, I will be ready to answer. I request you to listen the whole lecture carefully and previous lectures as well. Subscribe this channel, click the bell icon so that you can get the other notifications about my other videos. Thank you very much.